Statistical Mathematics. So a huge welcome to all of you and to all of our visitors, local to Bristol, local to Tokyo, and from outside. You're all very welcome. We have two talks this morning. The first from Dan Lawson. Dan is a data scientist here at the University of Bristol, and he's going to give us a talk on clarity, comparing heterogeneous data using dissimilarity. That's a very cool little title there. And I hope he's going to tell us more. We're going to try and have a 5-minute question and answer session at the end of each half. If you could type your questions in the chat, that would be great. And then we'll be able to return to them at the end of the session. So I shall hand over to you, Dan. Off you go.
Thank you very much. So hopefully you're now seeing my screen. So yes, so this is work that's been going on for the last couple of years. There is now a paper by the same name and software associated with this. So if you're interested, there is ways to follow it up. Okay. So the aim of this idea of comparing similarities is to provide a method that allows you to, in principle, compare anything to anything. Something that I think probably should get a name, transdomain statistical analysis. I'm not sure. It doesn't exist yet as a name. But so of course you can always compare anything to anything if you model the set of things jointly. But the idea behind clarity is to do that with minimal modeling assumptions. Of course, you have to start somewhere, and the place that we start here is by the relationships between entities in these two data sets, the anything and the anything. So the entities must be observed in two different domains. By domain, I might mean area of science, a thing that you can quantify, but I might just mean two observations of the same set of entities within one scientific domain as well. So hopefully that will become clear later on. The method works by taking a similarity matrix that is in one data set, decomposing that into something that we've called a structure and a relationship. And we do this for a range of model complexities. So what does structure mean? So we have a way of interpreting this structure, and it's totally analogous to a clustering, but it's more general than that. And so it also includes mixtures, but also positions in principal components space. And I think that leads to a relationship to the geometric data analysis for this series. So now we have some structure. We also have a relationship, and that can be interpreted as how do the clusters that we've just learned relate to one another. And then this notion of doing a comparison between one data set and another is asking whether the structure from one matrix predicts a second. Okay, so to give a visual abstract of what's going on in this method, I've got an example data set. This is all simulated data, but it explains what's the point of the method. So we have on the y-axis, we have a whole bunch of objects that we've observed. Here they are positions on a genome, genetic loci. And then we've observed them in some different domains. So we may have observed them in bioinformatics. We might have observed whether they are methylated. That means prevented from being read by the kind of cell's mechanisms of copying. And we've observed that for a number of different features. Here, people. And those people may or may not be annotated. Here they're annotated by whether they have a tumor or not. And so we would expect to see clustering here, and it's that clustering that we will call the structure. So the features themselves get hidden into similarity. We work on the similarity between these loci. And so this allows us to compare to a data set that comes from a different kind of measurement scale. Here it's expression data. So this is looking in the cell and measuring whether you find a gene, a protein that was transcribed from a particular bit of DNA. And as you can see from the kind of different color scales of these heat maps, these are very different in terms of how they quantitatively appear in the features. And modeling those features would be very hard, but the similarity structure is conserved across these. And you can see, look, we don't need the same set of individuals to be measured. So these are different feature sets. But what we can do is we can ask, is the structure the same? And here I made the structure not the same. I injected some genetic loci that had different structure, and I made these anomalies. And we are interested in doing many things. One of them is finding such loci that are not as expected in the second data set compared to what we see in the first. And so we quantify this with something called the persistence plot. This is not a persistence diagram that you might be familiar with from topological data analysis, but it is kind of inspired by that. So here we have now a model of different complexity, and we ask, when we have a model of a certain complexity, what are the residuals basically of each of these items, these loci? And these things persist if they are an anomaly. So that means that that the high residuals are present for all complexity. Okay, so I will explain all of that in more detail. So to interpret what we mean by structure, it's useful to think about mixture models. High-dimensional mixture models are popular in a huge variety of contexts. They're used in genetics a lot, and I care about their application there. They're used in graph theory a lot, and they are used in topic modeling for document analysis. Many of the viewers will be familiar with mixture models, and so these are typically simple when you have a setup that is talking about items on their x-axis here that are described by their features. So we have L features and we have N items. And it's simple because this matrix decomposes nicely into an N by K mixture matrix. So this is how much each item belongs to each of the K latent classes, and then the properties of those latent classes. That's X. So each of the properties defines its distribution on the L features. And so you can see there's a kind of localization that happens. The data in one position only depends on its corresponding row of A and column of X. And that makes the inference simple, and therefore this kind of works. When you do this inference, it just works and behaves itself. Things are a bit harder when you try to jump to a similarity matrix. So similarity matrix is an N by N set of similarities between the N objects. And we have the same matrix A, but now we have a matrix X, which is K by K matrix of the similarities between the latent objects. And so now if we're interested in the value of one of these similarities that we observe, there is a kind of loss of this locality in X, because it depends on all of the values of X, because you pick up your similarities through every possible membership that you have. And this is not very well explored in the statistical inference literature. So if you want to infer this, there are a couple of methods to do so. So some people here work on spectral methods to do that. These are not typically seen as inferential tools, but more theoretical tools. But although you can apply it, but there are some difficulties. the main tool that people have used is the mixed membership stochastic block model. This is focused on graphs, so the data tends to be sparse and present where there is edges, so 1 where there is an edge and a 0 otherwise. Now, if you apply mixed membership stochastic block models to the sort of similarity matrix I care about, it doesn't work. So I had a student work on this a couple of years ago. All he did was implement the published algorithm for mixed membership stochastic block models. It's quite a sophisticated algorithm. It uses an empirical Bayes procedure with variational approximation to quantify how good a particular set of parameters is. It gets you a whole posterior distribution. I think the literature considers this mostly done, but it doesn't work, the published algorithm. And the reason the published algorithm doesn't work is that in order to describe the distribution of the posterior parameters of such a model, you have to perform a search process as well as having a model for what's going on. And it's the search part that goes wrong. So the update rules that they use for empirical Bayes get stuck in a local optima, and specifically it loses components. So if we jump back to this picture, we end up with some columns of the A matrix that get set to 0. The corresponding rows and columns of the X matrix march off to some random arbitrary value, and then they're never recovered. So there is an additional search problem that needs to be solved. And Tjun got some way towards solving that through an ad hoc search procedure. But overall, we found this to not be a very satisfactory approach. So we instead, for the Clarity paper, came up with an alternative method. It's simply to try to minimize the squared residuals. So L is my loss function, Y is my similarity matrix for the first data set that I'm interested in, A is what I'm going to call structure, X is the relationship. So we just want to minimize this with respect to A and X, subject to the fact that A is supposed to be a mixture. And it turns out that you can do this with an iterative multiplicative scheme. So you first update A by multiplying it by this matrix here, and then you update X by solving for the X that best explains Y, conditional on X, and on A, sorry. And then you keep iterating, going backwards and forwards. And I will give you an intuition behind how these work. So the X update is quite straightforward. It's just linear algebra. So we've got this relationship. We want it to be Y equals AX A transpose. And therefore, we can just turn these rectangular A matrices into something that have an inverse. So we'll pre-multiply by A transpose A. That's now a K by K matrix. Do the same thing on the other side. And now we can write down the inverse of this matrix, which I called G. And therefore, this part of the equation drops out, and we end up with a solution for X. And this work, sometimes you have to use a pseudo inverse just in case you're dealing with incomplete rank data, but other than that, this just works. The A rule is more complicated, but it's still based on something that's now 19 years old. So Lian Sung described an algorithm for updating these non-negative matrix factorization problems. The trick here is to use an auxiliary function. So you write down some function that's not your loss function that you care about, but that you can guarantee that if you minimize it, that you move towards the optima of your actual loss function. So in their case, this was a quadratic because they had y minus ax is the loss. Here we've got y minus ax A transpose. So we've got a quartic in A. That makes the calculations harder, but actually their procedure just works. So the intuition behind how this works is you just do a gradient descent procedure. So you take a step in the direction of the gradient minima, but the step size eta ij is chosen to be such that the additive terms drop out and you only get a multiplicative term left. Where this strange equation comes from is basically you take the first derivative of the lost function, that gives you the numerator, you get the second derivative from the denominator, and then there is an additional term when you do those derivatives, which is this one here, and everything else drops out. So this is not very it's not very hard to implement, and it is guaranteed to be non-decreasing in L if from everything I've just said up to now. The problem is it doesn't respect that A is supposed to be a mixture. So in order to keep A a mixture, you have to normalize, and this final step, this normalization, doesn't guarantee that you've got a non-decreasing loss. In practice, if you initialize at A that is a mixture, it doesn't move from being a mixture very much, just due to numerical instabilities. And so in practice, this still works, provided that the truth is reasonably close to a mixture. So it is possible to find counterexamples where this whole thing stops working, but in practice it does work. As with all greedy optimization algorithms, you get stuck in local optima. But because we're interested in doing this for very many values of k, the complexity, number of latent mixtures, we can use one solution that we have to improve others. So basically, the published algorithm uses the k-1th solution, and it proposes a tweak to that for the kth solution, and then performs a local optimization. So we go all the way through up to from k equals 1 to k equals k max, and then we go backwards again. So this is just a way to make this sort of procedure work in practice to avoid the fact that we get stuck in local optima. In practice, what happens is that there's some range of k where it does work, and then some range of k where you get stuck in local optima that don't replicate. Okay, so why have we gone to all of this effort? The reason is that if the truth of some data set is reasonably close to a hierarchical clustering, then this allows us to perfectly capture that hierarchical clustering, and it allows us to capture richer true structures as well. So here I've generated a similarity matrix, that's this one, from a tree, and the tree, every branch on the tree induces a cluster. So this one here induces a cluster that is made up of these three kind of true clusters. And what we would hope to do is recover all of these clusters with our definition of what structure is, our mixtures, with our mixture model. And so when we do this, what we find that this actually happens. So this is with k equals 2. At k equals 2, the mixture that we recover is basically saying there is the pink cluster and there is the yellow cluster, and then you explain things that are neither of those clusters as a mixture. And as we add more and more components, we eventually converge on describing every true cluster as an actual cluster. And so so every single one of these kind of real clusters gets represented in this richer mixture representation. But the advantage is that if there were genuine mixtures going on in the tree, those would also be captured in this structure. And so this means we've got a kind of clear understanding of what we mean by structure. It's a kind of soft hierarchical clustering. So I like mixtures, they're interpretable. Unfortunately, they are not necessary for what I was trying to achieve with clarity, maybe fortunately. So if we instead remove the constraint that the A should be a mixture and just allow it to be anything, then we've got the same loss function, y is being approximated by A, X, A transpose. Of course, you can just use the principal components analysis in order to arrive at A and X. So A becomes the eigenvector matrix, X becomes the eigenvalue matrix, diagonal matrix of the eigenvalues. And computing this is way more efficient than computing the previous mixtures. And I'm not going to show it today, but there's kind of many kind of theoretical arguments as to why a mixture should end up being representable as a spectral decomposition. And some people here can speak to that better than I can. And also empirically, it really does. Okay, so in any case, however we arrive at our definitions of A and X, we now want to use them to compare to similarity matrices. So the procedure is that you learn A and X from the first data set, so that's Y1, so that's a similarity matrix, and we then keep that structure that we learned, the A, and then we apply it to the second similarity matrix, Y2. And here we learn a totally new X, but keep the same A. So we're really asking, how does the structure from 1 explain the data from 2? And then that's the kind of the hard bit done. The rest is just figuring out what the answer means. And unfortunately, that's not trivial either. But the model obviously outputs residuals. So that is how good the model fit is at complexity k, comparing items I and j. And persistences, which I showed in the picture at the beginning, this is just the sum of the squared residuals for item I across all of the things it can be compared to, all of the j, at a particular complexity k. So to show how this works, I simulated data under a tree, and then I rescaled the tree. So I'm generating data that has some structure, then I'm generating data that has the same structure, but with a different relationship between the items. So I'm reorganizing the relationship between the clusters. So this changes X, but not A. And then optionally, we can then change the structure too. So we take some items from a particular cluster and we add in some similarity with some other bit of the tree, effectively. And this changes A as well as X. So when we look at the residuals and persistences for this, this is our data that we learn on. This is Y1. And so inside each of these clusters, tips, sorry, is many items, so maybe 10 or so, or 20. And so we have the kind of average dissimilarity within each one scored here. Then we look at the persistences. So how bad is the model fit? So when we're looking within the data set that we learned on, of course, if you've got a low dimensional representation of this, then you get high residuals, so high persistences, but those are not statistically significant. So that's what the kind of fading out means. If we now do the same procedure on the data set where I've changed the relationships, the residuals are significant for low complexity, and that's because how important the various directions in this space are, the various clusters, has changed. So as you can kind of see, the biggest split at complexity 1 here was between these clusters 1 to 3 and everything else, whereas now that's not the largest split anymore, and so it focuses instead on T6 through T10, something like that. But these do not persist. So these changes in relationship do not persist, and therefore when we get to some complexity, here it's 6, then the residuals stop being significant anymore, and we're left instead with saying that this structure is explained. But when I insert some items that have a mixture, so here I've taken these items in T2, I've made some of them be a mixture from T9 as well, and then the residuals for T2 of course go up hugely, and the structure of that residuals tells you how the matrix was changed, how the data were changed. So it tells you that T2 is the kind of recipient of this change, but you can look it up and you can figure out it came from T9. And these residuals persist. So we're looking for persistent residuals when we're looking for a change of structure. Okay, so this is not geometric, but I can make it related to that, I think. So X2 is currently unconstrained. The structure, that means the structure is the same and the relationship can be arbitrary. As a consequence, the interpretation of these kind of complexities is whether the low dimensional subspace that is spanned by the first k columns of the eigen decomposition of Y1, is that aligned to the same thing in Y2? And so it will be, the persistence is low if they are aligned and high otherwise. We'd like to measure how similar the x's are, so how similar are the relationships, and potentially constrain them. Now, as well, this process of having a different relationship can also change topological measures. So if we were to make a topological, like an actual persistence diagram of the clustering of A, or the positions of A. The traditional thing in TDA is to do this with a ball. So if you're instead looking at the version of this that's been rescaled, where all the directions have been rescaled by this X2, then you can think of that as either moving the points about, stretching all of the directions, or as using a non-spherical kernel around each of these points. And that could induce a different topological structure. So I haven't looked into that, but I think it's interesting. As an aside, statistical significance is a right pain in the bum for these sorts of questions, because you can't bootstrap similarities. So similarities are all correlated because the value ij is correlated with the value ij dashed because it's computed from the same set of features. There's a random subsampling procedure that works, that we use. And I should say, we're also interested in doing this when there are no features to the data. So many of the questions I have in genetics, you can create a similarity without having features. And so then you've got to figure out what the independent structure is. There is some theory, and I'm not going to spend any time on it, except because Vinesh can explain it better than I do. But I can tell you, broadly speaking, what it means. So what it means is that if there were some true structure, these are the unprimed similarities, but we observe only noisy versions of the similarities, so that's the primed versions. Vinesh was able to prove that the difference in Frobenius norm that you get by applying our entire procedure is bounded. So so this is this is the Frobenius norm that you'd get. So you can see we're working with the x2s here. So we've learned with y1. We're learning the prediction onto y2 here, and he showed that if you had the true structure, you would get something, and if you get the kind of statistically noised up version of the data, then you don't make a very large mistake in that prediction. So that means that this definition of structural comparison that we've chosen to do is a statistically meaningful thing to be looking at. Okay, a quick whiz through some applications. So the first is comparing genetics to language, the second is within linguistics, and the third is culture and economics. So comparing genetics to language is a very hard problem because these things are sort of a little bit tree-like, but actually very far from tree-like. So if you look at language similarities, this is all North Eurasian languages, then there is a kind of strong clustering structure along the diagonals. That means that within a language subclade, there is a kind of there is a clade, but it's not perfect. So there are kind of off diagonal red parts, so high similarities. So this is quite tree-like. The genetics is much less tree-like, and it's the similarity matrices that you get are not visually really very similar. So we would often it would be hard to see exactly what's going on with these two things, and there's no methods that would allow us to do this comparison properly. However, if we learn the genetic similarity and then predict the linguistic similarity, we find that we get this reconstruction, top left, of the language data, which is the top middle. And you can probably see that these two things are extremely similar, despite the fact that the genetics data doesn't really look like that. And that's because it has the same clusters in, or the same soft clusters. Specifically, it has the same mixtures. And looking at the residuals is very informative, so I'm going to zoom in on that on the next picture. So this is the residuals for that matrix, and everything where the residuals are high makes perfect sense. So the first is Hungarian. So Hungarian is a Finno-Ugric language that is spoken in Hungary and is very different to all of the other languages around it. And this sticks out very strongly because genetically Hungarians are not very different from the people around them. A similar thing happens with the Romance languages, Spanish, Portuguese, Italian, French, and Romanian. These all have a linguistic clustering that is not present in the genetic data. So there is obviously genetic structure that these populations share, but they share it with many other populations too. So everywhere the Roman Empire went, in some sense. So these structures all totally make sense. Another one that's worth highlighting is Greek. So there are Greek speakers in Greece and Greek speakers in modern day Turkey, and these of course speaks the language that's very similar, but genetically the Greek speakers in Turkey have become very similar to the Turkish people. The linguistic comparison is in some sense less stark. So this is comparing how words sound compared to what they mean. And if you look at these two matrices, you probably struggle to see any difference between them. I don't want to spend too much time on it, but what we find is that there are structural differences between these, and these all make sense too. So if we're trying to predict what thick words mean from what they sound like, we find there's some failure to predict at these low complexities. So that means the relationship has changed a little bit, but these disappear, and you just get left with this kind of wide scale, significant but not very clear persistencies. And that means that the matrix has changed in a way that is quite subtle, and it fits back with our simulation. Everything becomes persistent if only one small change is introduced. In looking in the other direction, we can see where these changes are. We see persistent changes in a few places, very localized places. And so the most striking of these is in the Ben and Hin languages, which you'll have to, I can only apologize that I don't know which languages they actually are, but I do know that those are Indo-European languages that are spoken in Central Asia, probably in India, whereas all of the other languages are Indo-European languages. So these are standing out, as are certain clusters in Central Europe. And again, the linguists tell me that this makes sense. Finally, comparing culture to economics. So here we've got a dissimilarity matrix of culture that was constructed from the World Values Survey. So this is asking you questions like, how happy are you to live next to somebody with HIV, live next to somebody who's gay, what's your feelings on war, all sorts of things. Are you happy? And it's a very well conducted survey. We also have data on economics taken from the World Bank. This is things like GDP and inequality. And what we find is that, of course, you can't totally predict culture from economics, but you can do a reasonably good job. And so the persistencies are all significant, but they stick out in certain places. So where do they stick out? So they stick out in Andorra. So it's not too surprising that the economics of Andorra is weird compared to its culture. So it's culturally very similar to the places around it, but it's economically much more, the money is much less fairly distributed because it's used as a tax haven. Vietnam is culturally much further from everybody else than is expected. That could just be because our ability to predict it from the economics is very low. Jordan is similar. And then jumping down to the examples at the bottom. Places in South America, such as Uruguay and Puerto Rico, as well as places in the kind of outer ring of Europe, such as Malta and Czech Republic, these are much more culturally similar to Europe than would be expected on their economics alone. Algeria is in the opposite direction. It's further from Europe. So looking into all of this, it all makes sense, and it allows us to draw inferences and form hypotheses that can then be tested with follow-on analyses. Okay, that's the end. There is an R package. You can just download. It should be very easy to use, and if it's not, let me know, as well as a paper that you can read. I'd just like to highlight some opportunities. So I've been talking about explaining one data set from another. That means that we're kind of using the language of regression, and I'd like to get to the point where similarities can be used in an actual model as well, in the same way that regular random variables are for regression. There's loads of application areas. There's loads of different methodologies that achieve the same sort of goal. The theory really needs some fleshing out, and I think that there's potential for geometric approaches to this as well. Okay, so that's me done. Has there been any questions in?
Thanks, Dan. Yeah, we do have a couple of really good questions from Patrick and Kenji, but I think we're overrunning by quite a chunk. So what I'm going to recommend is that we capture the information on the chat, and we share that with you so that you can continue the conversation offline. I hope that's okay.
Yeah, no problem.
So thank you very much. That was great. And we're going to move on to our second talk, which is by Tjun Yee Hoh, who recently joined the University of Bristol to research different types of structures present in high resolution microscopy data, amongst other interesting areas. And today giving us a talk on fibre analysis for super resolution microscopy data. So over to you.
Right. Okay, I hope I'm sharing that. Okay, so my name is Tjun Yee Hoh. Today I'll be talking about fibre analysis for super resolution microscopy data. There you go. So first off, what is super resolution microscopy and what does the data look like? So traditional optical microscopy is hindered by the fact that it cannot bypass the diffraction limit of light. So images, structures that are smaller than 200 nanometers in size cannot be resolved properly. Super resolution microscopy techniques bypass that limit. And one technique that we'll be using today is the single molecule localization microscopy technique, so SMLM for short. This method involves tagging small structures. In our case, we are looking at proteins of size around 3.5 nanometers. These proteins are tagged with fluorescent tags. Now, over time, these tags will emit light, and there are algorithms available to extract the molecular coordinates of these lights. So in the form of 2D coordinates. Now, the problem is over time, these tags may emit light multiple times. There may be multiple recordings of these locations, but there are algorithms to post-process this. And the measurement artifacts arising from this are summarized by localization positions. So to summarize, the data are simply two-dimensional coordinates. Each coordinate has an associated position. So what does the data look like? Here's an example. Here we have actin proteins, the location of actin proteins on the surface of human white blood cells. So here you can see the locations of the fluorescent tags. There is background noise that are not the result of these fluorescent tags. But as you can see, most of the fluorescent tags are clustered in a way that may suggest the appearance of fiber structures. So there has been previous work on the clustering behavior of these proteins. They've used Ripley's K function, paracorrelation, clustering maps, and a Bayesian approach with Gaussian mixture models as well. So clustering analysis has been relatively well studied. But then the study of fiber structures in these form of data is relatively understudied. So this is what we'll be looking at today. In particular, this is of interest to biologists because having fiber structures in the data suggests that there exists a fibrous cytoskeleton in the cells. This cytoskeleton forms a meshwork. You can think of it as a sieve of sorts. So it influences the behavior and movement of molecules around the surface of the cell. So the density of the meshwork prevents cells or molecules of certain size from entering the cell or exiting the cell. The structure of the fibers themselves, for example, the branching angles, suggest the presence of other molecules, other proteins as well. So interesting features in the structures may consist of the locations of the fibers themselves, the curvature of the fibers, intersection points and branching angles, and also the sizes and shapes of the enclosed regions formed by this meshwork. Okay, so how do we model our data? The way we do it here is we consider a hidden manifold model. So in the first bit, we have a uniform distribution on the region of interest, which simply governs the background noise. And the second part, we consider a distribution on a manifold M, given by W here. This manifold is simply a low dimensional curve. And since we are in the two-dimensional regime, this manifold M is simply a one-dimensional smooth curve given by the red line in the graph there. So we assume there's some distribution W on this manifold M, and this distribution is then convolved with a two-dimensional Gaussian distribution. And here on the left-hand side is a sample drawn for this hidden manifold distribution. Now, with this model, we could estimate the manifold M, but this is a relatively difficult problem. Estimation of manifolds give us at best a logarithmic rate of convergence, so that is problematic. And we consider instead an easier problem. We can resolve this by looking at the density of P, the density of this model. And if you can visualize that, the peaks, the local modes or maxima of this density, where the density is highest, will correspond almost to the red line. It will correspond almost to the manifold M. And these local modes or local maxima, we will call it the ridge of the density. So the ridge of this density approximates the manifold M. We also use the fact that ridge estimation is an easier problem. So it has polynomial rate of convergence instead. Okay, so what is a ridge? Here I give a more formal definition for the ridge. For a smooth function f from R2 to R, in this case, this function is simply the probability density function. I can denote nabla f its gradient, and H is Hessian matrix. If we take the decomposition of the Hessian, so we get the usual eigenvector matrix and the eigenvalue matrix. Sorry, and we order the eigenvectors according to the magnitude of the eigenvalues. So we focus on the second eigenvector. Then the ridge set can be defined as the set of points whereby the gradient of the density is equal to 0. But this gradient is projected in the direction of V2, the second eigenvector. So I have here the gradient of the density F projected in the direction of the second eigenvector. Now, ridge estimation has been relatively well studied. It has been applied in areas such as road mapping, earthquake fault line mapping, cosmology, where they try to reconstruct webs amongst galaxies, and also the mapping of blood vessels. And one established method is the subspace constraint mean shift algorithm, SCMS for short. Now, this algorithm takes in a probability density estimate. And given this density estimate, it iteratively finds the local maxima of this density. And in doing so, this maxima searching algorithm is constrained only to search in the direction of V2 at any given point. And traditionally, kernel density estimates of the probability density are used as inputs to this algorithm. Now, in our case here, I consider a Gaussian mixture model with added noise instead of the kernel density estimate. So why do I do this? Doing so allows us to account for background noise. It also provides an easy heuristic approach to feature detection for the fibers. And it also accounts for the precisions associated with the two-dimensional data. Okay, so this is the surrogate model that we use instead, using the Gaussian mixture model. Once again, we have the noise term, and then we have the Gaussian mixture model term. So pi are simply the mixture proportions, and phi here is the Gaussian distribution centered at mu k with covariance sigma k. So once again, for the sample that we've seen earlier, the sample drawn from the manifold model, we are able to fit to this sample the surrogate model here and obtain a probability density estimate. How do we do that? fairly straightforward. You can estimate the means mu k and the covariances sigma k of the Gaussian mixture model using the expectation maximization algorithm, or EM algorithm for short. Doing so, we can obtain a probability density estimate on the left-hand side here. For example, the black dots correspond to the background process, and the colored dots correspond to the Gaussian mixture components. So in this case, we have 4 estimated mixture components. And from this density estimate, we are then able to estimate the ridges. Or in this case, there's only one ridge governed by this black line here. So as you can see, it's fairly close to the underlying manifold M given by the red curve. Okay, so going back to the actual data sample that we had. So here's the actual data of molecular coordinates. On the right-hand side, you have the probability density estimate, in a sense, using the surrogate model. Here we have the various different Gaussian mixture components. And given these components, given that density estimate, we can estimate the ridges, which is given in red here. So these red lines, red dots correspond to the ridges. And as you can see from these ridge estimates, we are able to determine the curvature of the ridges, in a sense. We can determine where the intersection points are, what the branching angles are, and so on. So one problem with ridge estimation is that it is relatively computationally expensive. And there is also the problem that intersection points are not well defined for ridges. So we can turn to an alternative feature detection method. Here, this is thanks to the use of the Gaussian mixture model. By extracting principal components, we are able to do a fast heuristic feature detection for fibers. So we can extract the means to obtain the locations. We can extract the principal axis from the covariance matrices of the components. And in the left-hand graph here, we have the principal axis for the mixture components. Using that, we can perform some form of post-processing to obtain, once again, the fiber locations, branching points and angles, and sizes and shapes of the enclosed regions. Okay, and now I'll be moving back to the fact that each 2D coordinate has an associated localization precision. That is, each observed point has error involved with it. So at first, we had assumed that the true molecular coordinates come from the surrogate model. So the coordinates come from the mixture model. Now, we additionally assume that the observed data is perturbed by the localization positions given by SI. So here we have that each observed data is centered at the true molecular coordinate and perturbed by a circular Gaussian with covariance matrix SI squared. And thanks to the use of the mixture model in the surrogate model, the resultant likelihood is still in the form of a Gaussian mixture model. All that's changed is that we have the addition of the localization precision term, SI. And once again, we can obtain probability density estimates by using the expectation maximization algorithm. All we need now is just a deconvolution step in order to account for this additional SI term. Okay, to summarize the method that we have here, we have fitted a surrogate model involving background noise plus Gaussian mixture models. Using this, we are able to account for the localization precisions. We are able to obtain probability density estimates for this and perform ridge estimation. We are also able to perform heuristic feature detection thanks to the use of the principal components for the mixture using the mixture models. Okay, future work would involve possibly an alternative deconvolution step. So instead of considering the localization precisions that we have here, because we have access to the data underlying the fluorescent tag emissions, so we have time-wise information as to when the tags emit light, when the coordinates are captured. We can consider an alternative deconvolution step for this by considering this temporal behavior instead. Also, under certain conditions, ridge estimation is consistent, but we're looking to relax these conditions, mainly because the ridges, by definition, do not allow for intersections, or rather intersections are not well defined under ridges. We're also looking into more statistical methods for the feature detection for fibers, as opposed to the heuristic method that we have now. And that's it. And thanks for listening.
Great, thank you. We do have a question. I think we've got a couple of minutes. So from Song, is there a particular reason to use the Gaussian mixture model? If the data is 2D, KDE should work reasonably well. Is it an assumption of your data being mixtures of Gaussian?
Yeah, so in the original works using for ridge estimation, they use kernel density estimates. It works quite well. The ridge estimation is consistent for kernel density estimates. The main problem here is that we are unable to account for the precisions of the data. So we cannot account for the noise or noise error involved in the data using KDEs. And here we use a Gaussian mixture model, which is able to account for the precisions. And we believe that as it will be consistent as well as the number of components K increase as N increases. In a sense, it's a non-parametric estimation in the same way that the kernel density estimation is non-parametric.
So, sorry, just one quick follow-up question.
Yeah, go on.
Did you say precision? So the KDE cannot achieve the same precision that you want?
Sorry, by precision, I mean the each coordinate has a noise, has an error term associated with it. The kernel density estimate cannot account for this error term without doing some form of convolution, which is slower than what we're doing here.
I see. Okay, thank you very much.
In terms of the number of components, so we're hoping to have a consistent estimator using the Gaussian mixture model. So the number of components should grow as N grows. But yes, you can try to put a limit or lower limit in terms of the number of components, assuming prior knowledge, but this is not what we're trying to do here.
Okay, do we have any other questions from around the table? It's great to see so many of you here today. We do have a couple of minutes. Do we want to return to some of the questions that were being posed to Dan? I know there's been toing and froing on the chat. Are there any follow-up questions for Dan there? Dan, is there anything you'd like to go over in response to those questions?
No, so I think the chat has addressed everything.
Okay. Okay, great. Well, thank you very much, everybody. I think it's been a fantastic start to the series. Our next seminar, so make a date in your diary for the next one, is on the 10th of September, same time, 9 AM, 5 PM. And we welcome Daisuke Murakami and Ayaka Sakata to give another double-hander talk. So it just remains for me to thank everybody working furiously behind the scenes to bring this seminar series together, and in particular our colleague Kenji Fukumitsu and Song Liu, and our administrator coordinator Lily Rice, who set up the Eventbrite and has managed the morning today. So thanks everybody, really good start to the series, and I'll see you on the 10th of September.
Thank you. Thank you, Kate. See you next
Bye,
week.
Kenji. Nice to see you. Bye.
Bye.
Bye. Bye.